lecture will take us all the way to Antarctica, where some of the most extreme, deepest diving seals live. Weddell seals dive deep into the ocean to hunt for food while holding their breath for long periods of time. In order to do this, their physiology is highly specialized compared to other mammals, as their hearts and brains are protected from hypoxic injury that would be detrimental to terrestrial species. Dr. Allyson Hindle and Manu are here with us tonight to share their discoveries and what Weddell seals can teach us about new possibilities to treat heart attack and stroke, as well as their own physiological limits in a changing habitat. Now, a little bit about these lovely guests that are here. Dr. Allyson Hindle is an assistant professor in the Department of Anesthesiology at Massachusetts General Hospital, Harvard Medical School. She is a comparative physiologist who studies animals that live in extreme conditions. She tries to identify the physiological limits of species within their environments, which can tell us how they may be able to respond or not to changing habitats, and to provide insights for human medicine based on nature's solutions to physiological challenges. Little mouthful there. Now we also have Dr. Manu. He is also an associate professor in the Department of Anesthesia and Ophthalmology at Massachusetts General Hospital, Harvard Medical School. He is a translational molecular biology and physiologist with extensive cross-disciplinary research expertise and 18 plus years of experience generating and characterizing preclinical animal models of human disease, including cardiovascular disease and glaucoma, for target discovery, target validation, and developing therapeutics. Now, they also brought someone else with them, and a longtime friend of the Aquarium. She actually used to work at the Aquarium for a really long time. We're joined by a special guest, a science artist, I can talk, Maris Wicks, who has been a friend, like I said, of Aquarium. She's just really cool, so you will definitely want to check her out. So please join me in welcoming Allyson, Manu, and Maris.
Right, so also welcome from us, and thanks for being here in such a beautiful spring evening in Boston. Instead of going out on the Godzilla boats right in the harbor. I don't think I need to spend too much time introducing the three of us, so I think Maris and Allyson and myself have been beautifully introduced. Quite good. The star of the evening, of course, is the Weddell seal. And as you will hear throughout this presentation, the reason we're studying this animal is because it has this amazing capacity to dive and to stay submerged for a really long time. The longest before it dives, the Weddell seal is up to 90 minutes. And they actually exhale before they dive, so it's a amazing feature. But before we go plunge in the southern oceans and look at Weddell seals, I just want to talk for one slide about where Allyson and I work. So we work at the MGH. Who has been to the MGH? I'm assuming most of you, other than the people that I know that work there, have been to the MGH to see a doctor or to visit a sick relative or a friend. Healthcare is actually only one of the three missions of MGH. It's also education, and MGH actually was the first teaching hospital to be affiliated with Harvard Medical School. And the third pillar of the MGH goal is basically to do research. And research started 2 centuries ago. And one of the initial and probably most famous accomplishments that happened in MGH was that basically anesthesia was developed and discovered. So this is a picture of Dr. Warren removing a tumor from the jaw of a patient who was anesthetized by ether. And so when the patient woke up, he didn't remember anything, and there was no pain, which obviously was a very big advancement over what people were doing before. Currently, MGH is the largest hospital-based research program in the United States. And in fact, one of 3 doctors, medical doctors, do research. So there's 3,000 physicians at MGH and 1,000 in the research. There's 8,000 people in total who actually do research at MGH. It has a huge research budget, which is important because in these days of political turmoil, when people are talking about cutting the budget of the federal government for research, Boston would be hit very hard because we receive a lot of money from NIH to do research. And we're very successful actually. So this is just last year 620 patents were filed. And over the last century, 9 MGHers received the Nobel Prize, including Jack Szostak in 2009. So, I, for my entire adult life, have been studying this one very specific molecule called nitric oxide, which is a colorless, odorless gas, and was first described in the 18th century. And between then and now, we've learned that this molecule is involved in a whole host of physiological and pathophysiological functions in the human body. It's involved in blood pressure regulation, cardiac function, it's an important neurotransmitter, and it's an inflammatory molecule. In 1998, these two gentlemen, Furchgott, Hindle, and Manu, actually received the Nobel Prize for discovering that nitric oxide is a molecule in the vasculature that causes vasodilation. And there's a so over the past century, actually, drugs that generate nitric oxide, for example, nitroglycerin, have been used in the clinic to treat cardiovascular disease, including angina. And there's an interesting story around that, in that Alfred Nobel, who made his fortune using nitro to develop dynamite, was later on in his life diagnosed to have angina. And his doctor prescribed him nitroglycerin to treat his angina. And he famously said that it sounds like the irony of fate that I have been prescribed nitro internally. They have named it trinitrin in order not to upset pharmacists and the public. I also want to point out, Maris, our science cartoonist slash illustrator slash book writer slash explorer, so she has provided us with a number of cartoons which you will see throughout this presentation, and I love the cartoons. So nitric oxide, it's a very ubiquitous molecule. It's produced by cigarettes, and I don't know if you can read this, but actually these were anti-asthmatic cigarettes, of course developed by the French. It's also produced in combustion engines and by lightning. And actually there's one of the co-PIs on our NSF grant that allows us to go to the Antarctic is developing a device that takes advantage of the ability of lightning or sparks or plasma to develop nitric oxide. So that's another example of the cutting-edge technology that we developed at MGH. But I want to talk about where nitric oxide is produced in the body. So imagine for a second that you are a blood vessel. Again, one of Maris's cartoons. If you go inside the blood vessel, so this is where the blood flows, this is the lumen, there's a layer of endothelial cells. And surrounding that layer are the smooth muscle cells. So these smooth muscle cells can relax and contract and thereby can increase and decrease blood pressure. Nitric oxide is produced by these endothelial cells. And when they sense that blood pressure is too high, they release nitric oxide, and it's a gas so it can easily just diffuse into the underlying smooth muscle layer, which then relaxes, the blood vessel relaxes, the lumen becomes bigger, blood pressure drops. So that's how it works in terrestrial mammals. This is a somewhat boring part of the slide. One of the most important downstream targets on the molecular level of nitric oxide is an enzyme called guanylate cyclase. And this enzyme, when it's activated by nitric oxide, produces a molecule called cyclic guanosine monophosphate, or cyclic GMP. And it's the ability of cyclic GMP to activate other downstream proteins and kinases that actually allows it to cause phase relaxation and to cause smooth muscle cells to relax. Now, cyclic GMP is not only, or the level of cyclic GMP is not only controlled by the enzymes that make it, such as guanylate cyclase or other types of enzymes that make cyclic GMP, it's also controlled by enzymes that can destroy it, and these are called phosphodiesterases. And you probably heard about one of them, which is phosphodiesterase 5, because that's the target for CRS. I don't see too many. So if you know what it is and you use it, you know what it does. If you don't know, you don't have to know. I'm looking at my kids up there. So So what I've been doing for the last, as was mentioned, for the last 18 years is basically study this whole pathway as a potential therapeutic target for a whole host of diseases. And what we've done in the lab at MGH is develop mouse models of cardiovascular disease and glaucoma, and then we try to use activators and stimulators of this pathway to treat these animals. For today, I'm not going to talk about mice. I'm happy to do that some other time. We're going to talk about seals, and we're going to talk about how we're developing seal larvae. Not really, but also one of Maris's cartoons. And in the next couple of slides, Allyson will tell you a little bit more about the science we're doing with these seals. And this is Allyson's plan.
Thank you. Okay, how am I doing on sound? Can you hear me in the back?
Sounds good.
Better? Okay, great. So these are our seals. I know that there's been lots of amazing work done on this species of seal over decades, and that's one of the reasons that we're excited to work with them, because so much of their basic physiology is known. So we're now in a position where we can capitalize on all of this foundational research to really ask a very specific question in molecular biology and try to understand what it is about the seals that helps them do what they do. So seals, as have been mentioned a couple of times, these animals are breath hold hunters. So imagine that you're off to catch a fish at the grocery store. You're going to jog there. So you're going to open your front door and you're going to start holding your breath. And off you go, jogging to the grocery store. Now you're shopping, you're picking out the fish that you'd like to eat, you're packing your bags, and you're heading back home. All this time you're doing this while holding your breath. So these animals have a tremendous ability to tolerate low oxygen levels in their bodies. And they're able to do that without having cardiovascular problems, without having problems in organs that we really think of as humans as needing oxygen. Hearts and brains are our two main focus. So one of the ways that they do this is by having a very, very pronounced dive response. Humans have this, other vertebrates have it as well, but seals just have a really, really intense dive response. And one of the things that happens when they start holding their breath and diving is that their heart rate drops. So you're seeing a seal heading down to about a 200-meter dive. These guys can dive a mile deep. They can hold their breath for an hour and a half. This is just a short little dive for them, 200 meters deep. Catching a fish at the bottom, and you can see its heart rate dropping from around 100 beats per minute to below 20. That's a pretty significant drop. At the same time, their blood oxygen is declining as they're going through this dive, and it reaches a saturation in their arterial blood of around 25%. That's about like being on the top of Mount Everest, where humans struggle to go without supplementary oxygen. So it's interesting, I'm a zoologist, I'm a marine biologist by training, and so I come at this from a conservation physiology angle. I'm really interested in the seals, so it's been so exciting for me to be able to partner with folks at the medical school to learn a little bit more about the clinical implications of these species. And I gotta tell you, we work with a lot of anesthesiologists, and they do not like these numbers that we see in seals. These are concerning numbers in patients, but these seals do this many times a day for their entire lives with no harm. So no talk about seals would be complete without gratuitous videos and photos of seals, so that's this portion of the talk. So these guys exploit the fast ice in Antarctica. This is the southernmost animal in the world, and you'd think that they were lying on a beach. Doesn't seem to bother them. So they use this ice to protect them. They're predators, leopard seals, killer whales, orcas. They're not good enough divers to dive in under the ice and find these cracks where the seals breathe. So the seals come in really far south towards the continent of Antarctica, and they're able to have their pups in relative safety. So this is a pup swimming, and you can see the mom. You can get a sense just right off the top of how graceful these adult animals are underwater and how not so graceful the pups are as they learn how to dive. So seals elsewhere in the world have a really short lactation period. The moms just fatten up their pups over the course of days and then they take off. Because these seals are so protected, they're able to stay with their pups for weeks and teach them to dive. So it's a really amazing opportunity to access these animals and to study them. So, as we've mentioned, we're excited about studying these animals to see if they might teach us about cardiovascular physiology, about how they survive without low oxygen injury, and how their cells operate under stress. They're also an ice seal, and although they live in Antarctica, where climates are shifting much differently than they are in the Arctic, they still teach us a lot about animals that exploit this type of habitat and can give us clues as to what might be going on with species in the Arctic as their ice cover is changing and their habitat is changing. And these animals in the Arctic are just, by comparison, so much more difficult for us to access and study. It's ironic, but going to Antarctica is actually much easier than going up to ice floes and trying to see Arctic seals in boats. So we know that these animals do amazing things with their heart rate. We know that they tolerate extremely low levels of oxygen, and we're interested in a real specific piece of that story, and that's how they manage their blood flow throughout their body. We're interested in the vascular physiology that distributes oxygen when it's very low and when these animals are underwater holding their breath and trying to conserve oxygen. We're interested in how they're able to fine-tune blood flow to different tissues so that the critical tissues are getting what limited blood and oxygen is there, and that other tissues are going without. And in terrestrial animals like us, we know that low oxygen hypoxia is a signal for endothelial cells to produce nitric oxide and to cause vasodilation so that more blood rushes to these areas that are low in oxygen. Now, if you're a seal and you're underwater for an hour holding your breath, that's all the time. That's all your tissues. You can't be having these signals operating in this on and off way that we understand it in humans. So seals provide a model for us to understand gradations in this response, potentially, just different ways that this molecular biology might operate. So this is data from 1979. I mentioned we've been studying these guys for a long time. This was not me. This is foundational work that's really launched this project from one of our collaborators in Ecopia and this grant. And what you see are differences in the blood distribution to different tissues of the seal's body when it's at rest and when it's diving. And so what I want you to see is critical tissues like the brain, they're actually getting more blood, more of the blood that relatively is circulating through the body when they're diving. The heart circulation stays the same. The coronary arteries are getting kind of similar circulation even when they're diving. But tissues like the muscle, the spleen, the liver, the kidneys, these visceral organs that maybe are not super necessary when the animals are diving, those are seeing less blood. So there's differences in different vascular beds. Arteries are reacting differently. It's not this on or off situation the way we understand it in humans. So I need to put our study in a little bit of historical context because I think it's really important to talk about how these data are collected and talk about the research methods that we use, have used, would like to use, how this is an evolving conversation. And I think it's important that we challenge ourselves when we're working with live animals to continue to have these conversations and continue to do better. So these data are collected in the 70s by basically putting a seal in a bathtub and keeping its head underwater while you collect these measurements. We don't do that anymore. But we want to see how these animals act in their natural environment. We want to understand the effects of exercise. They're not really exercising in their bathtub. We want to understand the effects of pressure. All of these things limit our ability to take measurements. These animals are going somewhere where we can't follow to observe them. So how are we able to do that? Over time, technology has really been the savior of this type of research and allowed us to really expand what we know about these animals. So now we just make seals look really angry by gluing things to them like heart rate monitors. And in Antarctica, you can glue these things to an animal, and I mentioned they exploit this fast ice region in the southernmost, close to the land, and their predators don't follow them there, so they hang out during the breeding and the pupping season for several weeks. So you can go back out, you can find the really expensive seal after it's been out doing seal things for a couple of days or a week, and you can take your computers back. All of this data, this physiological data, we recorded at a really high frequency. Heart rate is really high resolution data. We just can't use satellites to transmit this data back to ourselves. So thankfully, these marine mammals grow a new coat every year. So all of the little marks, we just glue stuff to their fur, and then we take the stuff off, and then the little glue bits just fall off each year when the animal molts. So we've been able to really transform how we think about these animals and how we think about what they do in the ocean by changing how we ask questions about them. And so the next question as a scientist, I think, is important to ask is, how is the data different? So it's mostly the same. What it turns out is those forced dives, where animals were diving in the bathtub, really represents the extreme limits of their physiology. They are able to do that, but they don't do it all the time. So this is liver blood flow. On the top is the amount of blood that flows to the liver when the animal is diving versus on the surface of the bathtub. So really tiny little blue line, not much blood when the animal's diving in the bathtub. And really big gray bar, lots of blood going to the liver when the animal's at the surface. A real seal diving in the ocean has blood going to its liver almost all of the time. So there are some differences. On the other hand, kidneys, whether you're swimming in a bathtub or you're swimming in the Southern Ocean, you need to turn off blood flow to your kidneys. They're not important. If you're a seal and you're underwater, you need that blood to go to your brain. So by comparing and cross-referencing these multiple types of data sets that have been collected by different technologies over decades, we really start to get a picture of the physiology of these animals and how they're able to live in this extreme environment. So I guess I get the science part here. So I got to tell you about our hypotheses related to nitric oxide, why we care about seals, and why we're interested in studying the vasculature of these animals. So using our modern methods, what can we do? We can do lots of stuff in the laboratory. We can look at the molecules, we can stimulate them, we can see how they react under different types of conditions, but we need tissues to do that. So how do we go to a pristine place like Antarctica and collect tissues from seals? Well, we have to go basically and wait around for seals to die. They're wild animals, it does happen, and conveniently they die in a freezer. So we're able to sort of wander around and look for animals that have died of natural causes in these breeding colonies. Females die from complications due to pupping. Pups die for various reasons. They just fail to thrive. So we do have a reasonable source of tissues that we can collect and take back to the lab here in Boston and study. So we have these two questions about nitric oxide that we're trying to answer in these seals. I mentioned that in terrestrial animals, we sort of understand this as an on and off situation. So in seals, it can't be that simple. We think that hypoxia can't signal nitric oxide in the same way in seals because all their tissues are hypoxic all the time and the blood can't go everywhere. It needs to be controlled. So we think that nitric oxide induced vasodilation might be blunted in seals, and we're really interested to know if that's true and how they do it. So to get at that, we want to compare the activity of this main enzyme that responds to nitric oxide called guanylate cyclase, GC. We want to compare the activity of GC among different mammals, including seals. We also think that we would see a nitric oxide pathway difference in different vascular beds in the seals. So that brain that we know gets lots of blood and lots of oxygen when the animals are underwater, maybe this pathway operates differently in different regions, and that might be a way that seals control blood flow through their body. So we want to compare different vascular beds in the seals and look at the GC activity and the elements of the nitric oxide pathway in different tissues. So for today, I'm going to tell you just about a two-way comparison, comparing the carotid artery that feeds blood to the brain and the renal artery that feeds blood to the kidney.
But before she does that,
Just trying to keep you on your toes.
I'm stuck. I'm going to tell you a little bit about where we went to do this research and how we got there. So this is the world. On the bottom is this big, icy, windy continent called Antarctica. And that's where we went. And it's quite a trip to get there. So we started in Boston, and then we took a flight to Dallas, and then we flew from Dallas all the way to Sydney, Australia, and that's a 17-hour flight. It's one of the longest flights apparently you can take. But very comfortable plane. Even for somebody my height. Then we fly to Christchurch, and then from Christchurch we take a military plane down to the continent. And in Christchurch is really where the adventure started, I felt. So the first thing we do is we go to the headquarters in Christchurch of the US Antarctic Program, or USAP, where we get our first orientation, where they teach us about how to keep Antarctica pristine and how to be safe. And we also go to the CDC, or the Clothing Distribution Center, where they provide us with ECW, or extreme cold weather gear. And I'm going to use another one of Maris's cartoons to explain. So here's our seal friend, and he has blubber all over the place. Obviously, we don't have that. So what we do get from USAP, the CDC, is the iconic big red goose down, it's Canada goose, I guess, parka, which is really, really warm. You get snow pants, these bunny boots, which are really nice warm boots, goggles, a gaiter, gloves, hats. So these are the basics you need to actually survive while you're on the ice. So then we get on a military flight. This is a C-17, right? Sometimes we fly on a C-130, or if you're lucky, we fly on a regular Airbus, which is the most comfortable way of going. So this was the first time I deployed to Allyson. Alyson's been there 7 times. I've only been there twice. Maris has been there once, but for a really long time, like 3 months. So it's just a huge plane with a lot of cargo and people flying to the ice. And then you fly, and you fly over this amazing continent, and you fly over pack ice, and you fly over Antarctic mountains and large glaciers. It's incredibly beautiful to experience that. And then you land on the Ross Ice Shelf, which is the largest ice shelf on the planet. The ice is hundreds of feet thick, and that's the ice we do not want to melt from climate change, if you believe in that sort of thing. And then you get off the flight, and the first time I got off this plane, it's like landing on the moon. That's how it felt to me. It's an incredible experience. You just walk up this plane, you walk on the ice, and you're in Antarctica. And people are taking pictures, of course, lots of pictures. Right, and so once you're in the ice, so this is approximately 10 miles from the station where we have our lab and we have our dorms. And so then you get taken in these very large wheeled people transporters. This is by the way is Caitlin, and I think Caitlin's parents are here. And so what is this called again, Allyson? What is the name of the truck? It has a name, right? It's a dress. Yeah. Takes you to McMurdo. Okay, so again, let's orient ourselves. So Antarctica, there's approximately 50 or so bases all across the continent. The US has the largest one, which is called McMurdo. It also has one at South Pole. And something I just learned from Allyson is, since the Russians couldn't have South Pole, they put a base at Vostok, which is apparently the coldest place on the planet. You should send John there. So when we went to McMurdo, which is at the edge of the Ross Ice Shelf, and it's actually on an island called Ross Island. And what you see here is, so this is the island. This here is Erebus, which is the most southern active volcano on the planet. This thing here, what you see is a glacier coming off the island into the Ross Sea. This is the edge of the ice shelf, so this is permanent ice, and then every year this part of the ocean freezes, and this is where we actually collect our samples from the seals. And then on the peninsula, all the way on the top, there's two bases. There's Scott base, which is the Kiwi base, and there's McMurdo, which is the US base. And this is a picture of McMurdo, so I don't know if you see, but this is the lab, these are the dorms, this is the central building where there's 24/7 pizza. There's even 2 bars, but I forgot where they are. There's a hospital, the red building, and then there's a $2 million gallon tank of fuel that should last for 2 years in case the tanker that comes every year, icebreaker, doesn't get to the station to deliver a new fuel. Luckily, we don't look like this anymore when we're on the ice. So this is 100 years ago. This is actually Shackleton when his ship got stuck in the ice. And people, they traveled by dog sled. Now we travel by skidoo. And so this is what Allyson was talking about. So once you get down there, the ice, which is a long trip, but once you're there, the animals are actually very accessible. So this is Erebus. You see the smoke coming out of the top. So we're driving on skidoos. We're putting our gear behind us on a sled, and here are seals. And it's hard to see, but this is a whole tidal crack, and it's where these cracks forms that the seals can come out, and where the moms deliver their pups. And so this is pretty much our commute every day. So we wake up, we put on the gear, it takes a while, and then we go out and we go look for seals. The sun is up 24/7. There's this little circle in the sky, so it's always the light. We also use helicopters to go look for seals. So this is a helo flying over the edge of the seasonal sea ice. And I think later in the presentation you will see some orcas that are swimming at the edge of the sea ice trying to grab a seal. You see some seals here and here and here. So it was also the first time I was in the helicopters. It was cool. All right, and then of course, by far the coolest thing of being in Antarctica is being able to interact with the amazing wildlife that's down there. So these are emperor penguins. Again, you see Mount Erebus in the background. This is one of the Delbridge Islands, so every time there's frozen ocean water and there's an island, cracks form, so that's where the seals again get out. These penguins, this was a day where we were skidooing, doing our work, and suddenly we saw this line of 50 or 60 emperor penguins just waddling behind each other, just like in the movies. And we stopped, and they walked over, and they came to check us out. And we just said, it was a very windy day, that's why I turned on the sound. They're just walking around, checking us out, This is actually Caitlin and Allyson. Just a magical moment. And then these guys, these are the Adani penguins. So you see the seals all around the penguins. This is a Weddell seal pup, just a couple days old. As I also mentioned, they have no predators when they're on the ice, so they're not afraid at all. The pups, when they see you, they just kind of scoot over. The moms typically don't like that, but as long as you stay away from the mom, you're fine. This is the only seal that we named. His name is Edgar. And we named him because he kept going back to the same breeding hole. This is actually a diving hole in a diving hut where scuba divers get access to the amazing waters underneath the sea ice. And you can tell that he's been in a couple of fights. So this was the time of year when the animals start to mate, and the alpha males control the breeding holes. And if you lose, you lose, and you have to find another way to come to the surface. And Edgar found this dive hole. In the meantime, the moms are on the ice feeding their pups. This is actually a pup whose mom has either gone fishing or just left him and he's on his own. If he's strong enough, he survives. If not, not. So this is drinking. This I just want you to hear. This is an underwater video from one of my colleagues, Louis Hofstad, who was part of our team. And he just dunked his.
Cool.
That's so amazing. There's another one. The sound effect. The sound like spaceships. This is mom. And she's waiting for her pup to dive in. This is also the most amazing sound. So this one time we were on the ice working on a seal and we heard this noise, this high-pitched kind of zzz, and then followed by this big boom through the ice. And I was sitting and I felt it through the ice. It's incredible. Do I have more? No, it's Maris.
Hello, test.
I didn't turn it on.
It's on. I think I'm just muted. Hello? No? Just give me that. I'm just kidding.
It's kind of stuck.
If I just talk loud, can you hear me? Oh, kind of, sort of. Oh, I feel bad doing this. Hi, everybody. I'm Maris. I'm answering the question, who let a cartoonist down to Antarctica? And I have to let you know, I blame the aquarium. I'm not a scientist. I have a degree in illustration, which would terrify parents usually when I was talking to them about what my background is. I have 12 years of cumulative education experience, so I was at the aquarium for 8 years as a program educator. Is it?
No.
Oh man, I'm sorry. I feel bad doing this. I'll try and be quick. But my main job now, I left the aquarium about 2 years ago to become a full-time freelance writer and illustrator, and my focus is science comics. So I think this makes it go, right? That way. I have illustrated the first book, Primates, and written and illustrated the other 2 books. It's kind of a passion of mine. I love science, and I felt like I was abandoning it when I went to become an illustrator, but that it turns out if you love something, it always comes back to you. So, like I said, I'm not a scientist, but I have this passion. So my job is to kind of get people engaged with science via illustrations or cartoons or comics. So, the reason why I say I blame the aquarium is for coral reefs. The book right there, I decided that I wanted to do some field research, and I ended up on a research vessel that the aquarium goes down and collects fish with, just because I'd never seen a real coral reef before, and I saw one, and then I came back, and I was freaking out, and I go, I need to go do more stuff like this, and I found out the next day that there's actually a grant that sends artists and writers to Antarctica called the National Science Foundation's Artist and Writers Grant, and I applied, I received it, and I went to Antarctica. 6 months before I went to Antarctica, I was at the aquarium, doing World Oceans Day, which is a big festival we have on World Oceans Day, which is June 4th. Thank you. And I have my table set up, I'm talking to people about coral reefs, and next to me are these lovely people. It's Allyson and Manu and Caitlin. And this is 6 months before I get to the ice. And I meet them, we talk about outreach and everything, and it's great. I didn't think I'd ever see them again. So I get down to Antarctica, been there for about 3 weeks. And my job is to go and get embedded with science teams there and study their work, and I'm actually working on a long-term comic book about life and science in Antarctica. So my boss sets me up with this team, and he's like, okay, you're going to go out with the SEAL team, and they're going to go out on the ice, and you're going to observe what they do. So I set it all up, and I I don't know who I'm going with. I see their names and everything, and I get suited up, get on my clothes, we're getting ready to go out on the snowmobiles. And I go, I think I know you guys from somewhere. You look really familiar. I know you're in the Boston area because of MGH, and they're saying the same thing. We can't figure it out. And then like 15 minutes later, we're like, we did it at the aquarium. Long story short, the Aquarium is a fantastic organization that kind of allows for this stuff to happen. And I've been to countless lectures sitting where you were sitting. This is the first time behind the podium, but it's a pretty magical place and a pretty incredible resource for the Boston area. So I know you all agree with me on that because you're here. So how do you make comics about science? Well, I learn as much as I can, and then I package it in a way that I think will be engaging for people who aren't scientists, or people who are scientists but maybe don't know about this. So this turns into that. These are my notes. I filled 5 sketchbooks of just drawings and all sorts of stuff. And Manu just showed you the videos with what seals sound like, and hearing that is just unreal. So I've got the seals kind of saying chip, 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 and all of the weird, yeah, that's my seal impersonation. We're working on that for a while. Thank you. All of the experience of being in this place and seeing science happen in real life. And I know science for the most part. You guys, your field research is just a small sliver of what the actual research is, but it's the part that is really engaging. Not everybody's going to get a chance to go to Antarctica. Actually, a very, very tiny handful of people will get to go. So my goal is to kind of capture that experience and then also share with you the science that's happening down here. And part of what's amazing is that the transparency of me being able to talk to these guys and go back and forth. We've met a bunch of times, we brainstormed ideas of what we could put in this presentation to either help deliver the information better or give you a visual that will help you remember part of that information. So that's mostly my goal. So the comic about nitric oxide and going inside the blood vessel, what they did is Manu and Allyson supplied me with some slides that look similar to just diagrams that you would have in like a science presentation or a presentation of research. And we went back and forth and we talked and sketched, and I came up with a sketch. After talking about it, we did pencils, and then with the language, we kind of went back and forth as well. So that's kind of how a comic happens in short form. The book I'm working on is over 200 pages, so it's going to be a little different, and I'm still working with these guys as well to put their research into comics form. But that's kind of where I come into play. And it's not just me. There's a lot of people out there who believe really passionately in science outreach communication, whether you're a scientist or you're an educator or you just are excited about that type of stuff. It's an important link to have there. So I think that's it. I'm handing it back to one of you. Here, you can have your, thank you.
This was both of them all.
It's my turn, it's my turn. It's my turn. Okay, so these are some of our seals. These are some pictures of us working with seals. So I told you that we wait for seals to die and we're able to collect great tissues from them. But of course, animals that have died from some unknown cause to us, that's a problem scientifically. You're never really sure if there might be some complicating factors or pathologies that are affecting the data that we see. So we thought really hard about how can we get fresh, healthy tissues to try to baseline some of our information. And so one of the best way we thought to do that was to take blood samples. That's something that we can do. It's relatively minimally invasive for the animals. So those are the pictures that you see. So we get to a seal, there's some sedation involved. This is a 1,000-pound predator. For their safety and for ours, it's just a little bit better if there's Valium involved in this equation.
For the seal.
For whomever, you can just use your imagination about who gets Valium. So we give these guys a big health workup. They're weighed and measured. We kind of make sure that they're healthy. We run their blood chemistry to make sure that they're within the normal specs for their population. And then we isolate platelets from their blood, because platelets, it turns out, have a lot of this guanylate cyclase enzyme that responds to nitric oxide. So we can study this in blood samples from seals and other mammals and try to just look at species differences that way. So that's back to that first hypothesis. Is nitric oxide less able to vasodilate blood vessels of seals compared to other mammals? So the main comparison, because we're studying arteries, and this just happens to be another species that we have access to get arteries from, are sheep. This is a food animal, so tissues are more readily available. They're available in Boston, and so a lot of our comparisons are seals versus sheep. So looking at a couple of different arteries, These graphs are showing you the ability of nitric oxide to stimulate this enzyme in vitro test tube conditions. Just under perfect conditions for this enzyme, what's the maximum ability of nitric oxide to make this enzyme go? And on the blue are one artery, the red are a different artery, but you're seeing seals versus sheep. And what I'm hoping that you're seeing is that the sheep dots are always higher than the seal dots. So there's a lot of different things. There's a whole bunch of different rabbit holes we can go down to understand how much of each little bit of enzymes are existing in each of these different animals and how that's different between tissues. But the basic story is the same regardless of what we look at. Sheep are higher than seals. Nitric oxide is better able to stimulate this enzyme in sheep than it is in seals, which is great. That fits with our hypothesis that nitric oxide signaling is blunted in seals. But we have what we call a two-species problem. I can tell you that seals are lower than sheep, but I can't tell you if that's because sheep are high or because seals are low. I want the answer to be because seals are low, but I can't prove that. So what we have to do is we need to look across more species and see if we can see trends, and that's where those platelets come in. That's a lot easier for us to get samples of from lots of different mammals and really do a comparative analysis. So the next thing I'm showing you is a blank screen, is our seal, our volunteer, giving his blood sample. And we're comparing the ability of nitric oxide to stimulate guanylate cyclase in platelets of different mammals. So you've got the Weddell seal on the left-hand side. We got samples from harbor seals. We got samples from stellar sea lions, and then other terrestrial animals, dogs, sheep, and humans. And I always have to apologize for the poor humans, the two people who were conscripted into giving their blood sample unawares as they returned to the lab after lunch. So we'd like to go back and redo those data with a little more care and hopefully reduce the variation in the humans. But I think you can see that these terrestrial animals have a higher NO activation of this enzyme than all of the marine mammals, not just the Weddell seals. The harbor seals and the stellar sea lions still have these low activation levels. So that's really great. Maybe this is pointing to the fact that, in fact, our hypothesis is true, that this response to nitric oxide is reduced in seals and maybe other marine mammals. It makes sense that they have similar specializations that allow them to control blood flow to different parts of their body as they all hold their breath to hunt fish. And what's even greater is that sheep are higher than the seals, but the seals aren't even that high compared to the terrestrial animals. So this is actually a really great comparison. If we're able to resolve differences between seals and sheep, those are kind of compared to other terrestrial animals, those sheep are actually on the lower end. So our second hypothesis is, are these things different in different tissues? Is this pathway really responsible for the differences that we see in blood flow, in perfusion to critical tissues like the brain compared to less important tissues like the kidney when the animals are underwater? So we want to compare the baseline activities of this GC enzyme. We want to look at how much nitric oxide can stimulate the enzyme's activity, and we want to look at the amounts of protein and the amounts of RNA and DNA that exist in the different parts of the body. So I need to remind you that we're studying seals. Yes, I need to remind you of that. Then when we're comparing renal artery to the carotid artery, brain and kidney, we see that there's really not a difference in the baseline activity of this enzyme. So that's interesting. That goes against our hypothesis that this pathway might be responsible for how much perfusion each tissue receives. But there's a whole bunch of different ways to look at it. And I want to just remind you of this little pathway diagram that we have. And specifically, I want to remind you the names of the things that are in it, right? So nitric oxide is made by this guy up at the top, NOS3. This thing here, that's the GC enzyme that I'm talking about, the activity of. It's got different subunits, so we can look at the abundance of all the different subunits. And then we've got, of course, you haven't forgotten PDE5, which is the target of seal Alice. So when we look at the amount of RNA for the GC enzyme, these are the different subunits of the enzyme. Again, there's not really a compelling and exciting difference between the carotid artery that supplies the brain and the renal artery that supplies the kidneys. But some other things are different. There's very little NOS3. There's very little amount of this protein, this enzyme that produces nitric oxide in endothelial cells. So that might be a real big reason why the whole system could be blunted in seals. There's just not a lot of this enzyme in there. But there's really not a lot of that enzyme in the renal artery. So that tells us it's possible that the carotid artery, one way the system could be operating differently in the brain compared to the kidney, is that the brain just can produce more nitric oxide. So that enzyme can see, even though the activity of the enzyme doesn't differ, the enzyme can see more nitric oxide and respond. On the other side of the equation, again, not nearly as compelling a difference as with NOS3, but PDE5, our enzyme that is inhibited by Cialis, is slightly higher in the renal artery. So not only is the brain potentially getting more nitric oxide in the arteries that feed it, but it's also breaking down the byproduct a little bit less. Breaking down the byproduct, the signaling molecule that actually causes the vasodilation, a little bit less. So, more pictures about us collecting samples from seals. So one of the other final things that we're really interested in doing is leveraging as many modern molecular tools as we can to come at this question. What's possible now in this century that wasn't possible decades earlier in the last century? So how are we able to even measure the amount of RNA in different tissues for different proteins in seals? We didn't until this project started. We didn't have the tools to do this. So the first thing that this project did was to sequence the genome of the Weddell seal. So that's a resource now that's available for us and for many other scientists to ask questions about the health of these animals, their physiology, their anatomy. Innumerable questions can come from having this genome resource available. And so what I'm showing you here, this is just my genome slide. These are the first mammals really to be sequenced. And so you're seeing a kind of complicated figure that breaks out the lineages of mammals into things like the rodents and things like the primates. And the Laurasiatherians are the blue lines. That's where the seals fit. And so seals are carnivores. So we have a lot of other carnivores that have sequenced genomes that we're able to compare them to see what makes a seal a seal. So one of the main focuses that we have in these genomic questions is comparing seals to dogs. Basically, seals are water dogs, so we want to know what's different between a carnivore that operates on land and a carnivore that operates in the water. And there's one last tool that we are really excited about taking into the future and being able to ask detailed molecular biology and clinical questions in the lab studying a wild animal, and that's cell lines. So we thought a lot about different ways, as I mentioned, to get samples from these animals. So blood samples were a great way to get a minimally invasive cell type and tissue type from these animals. But they also give birth on the ice. And so there's this moment, if you're fast enough, because things are freezing really quickly, you can collect placentas from these animals. They don't consume their placentas like a lot of other species. I think that's because they freeze to the ground almost immediately, so they just don't have time. And so there's all these beautiful arteries and veins and blood vessels in placentas that we're able to get samples from and study, and we use them to establish cell lines so that we can screen metabolic pathways. We can use drugs that we would use in human or mouse cell lines to really start to compare seals to other mammals and really bring them online as a model system that can teach us about cardiovascular physiology and how these animals can survive low oxygen conditions, holding their breath, low heart rates, without heart attack and without stroke. So these are just the summaries of the two hypotheses that we've talked about today, that this nitric oxide pathway really does seem to operate differently in seals. And even within the seal, there are regional differences in some of the components of the pathway that we think really shine the light towards how these animals are able to regulate their vascular physiology while they're underwater, while they're holding their breath. So we're sort of wrapping up here. I have to show you the obligatory. How do you catch a seal? Does anyone really know? That's how you catch a seal. They're pretty, it's for any of you who've done work with rehab programs or have had experience working with seals, this is ridiculous. You couldn't do this anywhere else on the planet Earth. There's no 1,000-pound predator that's going to let you come up and put a hood over its eyes and then say, "Oh, you caught me." And that's really all it takes. And of course, nobody's ever videotaping you the day that you do it like beautifully and you look all great and you don't make any mistakes and you don't slip on the ice. And so this is a little bit slower than some of our captures. But that's it. Now we've got Valium and we're good to go on the blood sampling and the health assessment for this animal. Would you like to finish up, Dr. Five?
Sure.
Oh no, apparently the seal's not done yet.
Well, I wasn't getting the value. Lots of gin and tonic though. All right, so before we show you another 4-minute video, I just want to thank a lot of people, but I'm going to do it really quickly. So as you can imagine, a project this scope, getting there and doing all the work and bringing all the samples back and getting all the samples processed, it takes a village. There's too many people to mention. I just want to point out a few people. So Annie and Anne are currently in my lab, and Alyson's lab, working up thousands and thousands of samples we brought back from the ice. Luis and Dan are our main coverage from Santa Cruz, and I'll get back to Luis in a second. The genome work was done by Kirsten Nimrad's DOS group over at the Broad. Rachel was a vet who worked with Jen Burns' group, who studies molting and energy metabolism in Weddell seals down on the ice. And Jennifer was kind enough to lend us Rachel for a couple of weeks so we could get our blood samples from the Weddell seals in a safe way for both the seal and for ourselves. Obviously NSF, who funded the whole project. This was an absolute privilege for me to be part of this team. So this is our team B267, so this is me, this is Louise from UCSC, Allyson and Caitlin, who's now, where is she again?
She's in Idaho.
In Idaho chasing squirrels or something. True story. And this is Rachel. So it was an absolute privilege to be part of this group. It was, we were down there for I think a total of 18 weeks over 2 seasons, and it flew by. It was fun to work, with such talent and very hardworking people. Who else do we have? Right, so we showed you a lot of videos and pictures. Here are the people who took them. Mike was a telco guy who did the telephones down there, who we met on the first time we deployed. The first day we actually were in Christchurch, and he remains a good friend ever since. He actually came to visit us recently. Joel, Louise, Henry, and John also made videos and pictures. Obviously, Maris, thank you so much for working with us and for having us with the outreach and for making these beautiful cartoons. And I'm looking forward to seeing many more and to work on your book with you as much as we can. Anytime you want to come visit us in the lab, please do. You're always welcome. I do want to mention Warren Zabel. So Warren is one of the co-PIs on our grants. And he's been down to the ice many, many times, as early as going back to 1970 something. And I think Allyson showed you one of his papers. It wasn't for him; he probably wouldn't be doing this work. So Warren used to be chief of anesthesia at MGH. He's still the director of the center where we have our lab, and he's also He was appointed by President Bush and then later reappointed by President Obama on the Arctic Research Commission Committee. And the cool thing about Warren, because of all his work, he was a glacier in Antarctica. And you know, Allyson, I don't know where it is.
Give me the pointer.
She knows.
It's right over here. This is a pretty amazing special place in Antarctica as well. This is the Vinson Massif, and Mount Vinson, which is on the edge of it, is the highest mountain on the continent. It's the eighth highest peak in the world.
Also, Allyson, I want to thank you for keeping me alive twice. That's also a true story.
I promised his mother that I would do that.
And this is a picture actually, so I told you that the sun is up 24/7 when we go down there. It's late spring. This was when we went down to the ice the first time. We were on the ice by late September, so very early in, or was it late, early spring I guess. So the sun was still setting. This was one of the last sunsets on the ice. It was beautiful. And with this picture, I want to thank my wife and my sons. Can you stand up? Hi, Ethan. Eva and Jan, for allowing me to get out and talk to the other side of the world and be away for 18 weeks and pretend to be in college again and having fun and doing this amazing work with these amazing people and these amazing creatures, and then being able to bring it back to all of you and talking about it. So thank you. And we have one more video for you.
This is just to finish up. I think we can't leave a talk about Antarctica without really highlighting again what a special place it is. The Ross Sea is considered the last pristine ocean system on Earth. And very recently, the Ross Sea Marine Protected Reserve was established by an international by an amazing international partnership between many countries that operate in the Southern Ocean. So fishing is now prohibited in a lot of the areas that these seals rely on. It's a truly amazing ecosystem. We see a lot of the apex predators. I can't watch this video without thinking that we are so much too close to those animals. But at the time, it seemed like a really good idea.
Yeah! 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 Ah!
Thanks, everyone.
Do you guys ever look into why they exhale before they do dives, and how that can contribute to like the pathway for nitric oxide buildup or anything?
The conventional wisdom is that they need to do that because it prevents them from getting the bends because they go so deep.
Okay. Yeah.
So they're sort of limited. It's sort of amazing that not only can they hold their breath that long, but they do it without their long oxygen store.
At what age do the pups start diving, and how deep do they go at those young ages? Do you know?
They start deep, a couple weeks old, maybe within 3 weeks, they're in the water, and they don't go very deep to start off with. And partly that's because these tidal cracks where the moms and the pups usually are right nestled up against islands, and so it's actually not that deep there for them to go. But the moms dive, they do these parallel dives as the moms teach them to go in the water, and the moms will not feed them unless they come and they do the swimming lesson. So they really start getting in there a couple weeks of age.
And it's really funny to see them try to get out of the diving board because they can't.
Were you able to measure blood pH, the acidosis?
Yeah, and it stays pretty stable over, this is work that others have done in the past. It stays pretty stable over the course of maybe a 20, 23-minute dive. That seems to be the limit of adult dive length that the animals really just use oxygen. After that, lactic acid starts to spike in the blood, and you see the pH change really sharply.
Do you recover quickly or not?
Sorry about that. The question was, what do we know about blood pH, and then how? And so for the first maybe 20, 23 minutes, everything is very stable, and it seems like it gives us all the signals of oxygen-based metabolism. After about 20, 23 minutes, they start to use anaerobic pathways, and you see buildup of lactic acid, and pH changes in the blood. But it recovers very quickly when they come back to the surface and they start to breathe oxygen again. The buffering capacity of the tissues in the blood is very high. That's a hallmark of divers.
Are you able to check their baseline health status? I mean, you had a vet with you, or do you do any immunology on the blood or anything like that?
The question is, how do we check their health? And that is largely done. A group that I was with a couple years ago published a paper on what are the baseline blood conditions for these animals so that we have a reference to work from. So we do basic things like a complete blood count. We do serum chemistries. We just assess white cell count to see if they've got infections on board or anything like that. It's really basic. A lot of it is just looking at them and having a good feeling about the quality of their coat and their blubber layer.
I was just wondering if there were any comparisons with other marine mammals like whales or dolphins, with any of the data that's been looked at, especially because there could be a captive dolphin type study or whatever.
Yeah. Does someone else want to answer a question?
You're the marine mammal guy.
I thought the marine mammal questions. The question is, so what are the comparisons between seals and cetaceans, particularly since there's a number of dolphins in captivity? We know very little about the big whales. There's just been amazingly few studies done on these animals, and they're not in captive situations. Dolphins are a little bit better studied, and a lot of these pathways do seem to be kind of similar amongst these species. We've been looking at expanding our comparisons into cetaceans a little bit as we go forward. But I think a lot of the bones of what happens are the same between seals and dolphins, but it's also a really amazing genetic question because these are two lineages that have gone back into the ocean from very different origins. So I think there's a lot of work to be done still to see whether we're seeing convergent evolution or different strategies.
that's a good question. And as always in science, you start with a hypothesis and you have this idea in mind and this pathway. Usually you think you're going there and you're going to end up right there. For example, the question is what could the application be for human health and disease from our work. If we, for example, understand how a Weddell seal regulates the expression of nitric oxide synthase differently in different types of blood vessels, we may understand more about how this the gene and the enzyme is regulated in general, and we could then target that new pathway or that new target to maybe enhance endocrine signaling in patients that have endothelial dysfunction, which is a very common cause of, for example, systemic hypertension. So that's one example. The other example is, if you were able to understand how these. So Allyson was telling you that when seals dive, despite all their adaptations, they still become very hypoxic for a really long time. If we understand how their tissues can withstand that hypoxia, we could potentially maybe do they express some sort of protective molecule, for example. Can we identify that, and can we then translate that to treating diseases in people that are associated with hypoxia, such as stroke, LMI, and certain types of cancers and certain types of inflammation? But we are really at the very basic end of the spectrum now. So between where we are now, doing the basic biology and microbiology and tissue collection, and getting to the clinical application, that's going to take some time. So.
Did you try to do any study on the diving people, like on free divers, versus people who don't dive?
So I didn't do it, but I think it has been done. No, people have they looked at these free diving?
A little bit. Yeah, the question is humans that hold their breath in free dive, what would be the comparisons there? The modern free divers have not been very extensively studied. That's just starting to happen now. But there is some interesting work done by Warren Sapel, our director in Kopiai, on the pearl divers in Southeast Asia, these lineages of women that have been doing this for generations. And they show they are, for humans, I mean, we have a range of ability that is much less than seals, but they're really on the seal end of the human ability. So it's neat to see human specialization as well.
Because oddly enough, actually, there is a relatively big freediving community in Boston.
Yeah,
so if you want to, they probably would people would want to.
I learned that when I moved here, and so I think it's a really interesting opportunity.
If you dive and you want to come by the lab, you have plenty of people to draw your blood. I was one of the 2 humans.
Yeah, great.
Let me go. The question is, do they have to take a break and pause between dives? Well, I guess yes, they do, and it also depends how long they dive. So on these short dives, for example, Edgar, the one that we showed you that came up in the dive hole, you could almost set your clock to him. He would dive for 5 minutes, come back, rest for 2 minutes, breathe in, breathe out, and dive again for a couple of minutes. I believe that if they go for the really deep, long dives, they have to recover longer.
Yeah, if they stay within this aerobic dive limit, we call it, this time frame in which they're really just drawing down their oxygen stores, they can replenish those extremely quickly. Just a couple of breaths and they can be back down to where the fish are. So it's pretty advantageous for them to stay in those shorter dive ranges. When they push it and they're building up lactic acid and their pH in their blood is changing, they need to recover longer at the surface. So it's a trade-off that they have to make to get to where the fish are.
How do you explain the exhale before the dive, and what type of underwater camera equipment is that?
I'll take a camera question. The one that I showed you, it was just a GoPro on a stick. Plug it in. I think some of the more beautiful pictures were done with a real nice underwater camera. Why do they exhale? So how do they exhale, or why do they exhale? So I think we.
Yeah, they exhale because they can't be exposed to nitrogen at depth. That puts them at risk for developing the bends. And so by exhaling, they get that out of their system, and it prevents them from having a gas bubble problem because they dive so deep.
Let's go all the way up top.
Two questions, and they're probably related, but I was a little confused with the monitors that you place on them, on the seals. What they were actually, what information you were getting from them, and then also, how do you think the in vitro studies compare to the real results you get if you were able to take blood samples at, say, 225 feet or something?
So there were two questions. The first one was what data we got from the monitor. The monitors with lipopurans.
Those, I can answer that quickly. Those are mostly photos from previous projects, and those animals that you saw were carrying EKG monitors so that we can track their heart rate underwater. That's the main one. They also wear a satellite tag so that you can find them again, and they carry a VHF radio beacon so that you can sort of isolate them. So the satellite tag actually sends you an email when the animal comes back to the surface, and the tag starts to transmit again. And then you have to go out on your skidoo with an antenna and listen for the beeping to find your radio collared seal, essentially.
And the second question was how do the in vitro results relate to what's really going on in life? And that's a more general scientific question. And we see it all the time. How does any of the work we do in mouse models relate to human health and disease? As scientists, we always use models to try to get to the bottom of very complicated molecular mechanisms. And then sometimes they are reproducible as you go up the organism, and sometimes they're not. We just started doing the cell work, so it'll be interesting to see whether, for example, cells we collect from seals, whether they're more resistant to hypoxia than cells we collect from sheep. That would, for example, reflect well with what is in vitro with what is going on in vivo. Yeah.
So I have two questions. One is whether the core temperature remains constant, and whether in organs or in areas like where the kidneys are, the temperatures are lower. And second, which also relates, is what happens in organs like kidney that get less blood flow, and how they sustain reperfusion agent, for example.
Two questions. I'm going to go to the first one again. What is the temperature in the seals as they die? Does it stay constant, or are there regional differences?
Yeah, I'm not sure I'm comfortable saying that I know the answer to that. There's different data that tells us some different things. I think largely they do maintain their core temperature the whole time. There's evidence that the exercising muscle, because it's usually isolated, the perfusion is isolated, they're not letting that lactate that's building up in working muscle get back into the central system, so the muscle gets hot. There's also some evidence that shows that some regions get cool. So I'm not really sure. I think it's a complicated story, and we don't know the answer yet.
And the second part of the question was about
how do kidneys survive reperfusion injury?
So that's exactly one of the questions we're trying to tackle, and we don't know yet. It's not only the kidneys, even their brains, they stay perfused, yes, but they are hypoxic. If our brains become as hypoxic as their brains, I can guarantee you I'm going to lose the rest of my body here. So that's why we brought back these tissues, why we have these stem cells that we can hopefully differentiate into brain-like neurons that we can then put on hypoxic conditions and then compare how they respond to how cells from terrestrial mammals respond. We have, as Allyson explained, we have the genome. We can look for genes in genome-wide fashion that maybe are associated with the ability of Weddell seals to withstand these hypoxic and ischemic reperfusion events. But that's one of these examples where it's going to take a long time, I think, before we get to the answer.
So are you illustrating your experience there, or are you going to illustrate like the whole research, like when it's done?
The question is about the Antarctica book that I'm working on and whether or not I'm going to illustrate just while I was there or everything else. Is that the so my proposal, actually the National Science Foundation Artist and Writers Grant is similar to a science research proposal. So I laid out basically a comic that will detail a bit of the history, what it takes to survive in Antarctica, and then me shadowing 6 major groups. I ended up doing more like 16 when I was down there, so I had about 2.5 months. So my goal is to have kind of like 5 or 6 core groups that I'm following up with while I'm off the ice. Obviously, these guys is one of those teams, and to kind of go deeper into the science of that. So this is, in the book, I'm really interested in looking at what's happening molecularly inside the seals and really kind of nerding out about that part. A lot of the other stuff in Antarctica, just talking about general geology, I spent time on Mount Erebus, the volcano. So it's kind of, it will lean bio in the bio area, but there's also astrophysics. There's a lot of atmospheric science stuff. There's a lot of climate change science stuff as well. So looking at ways to connect them, but also having the human aspect. I think it's a disservice to not talk about like my favorite thing to tell kids about and grown-ups is like you pee in a bottle when you're there because you can't actually pee on Antarctica because it's pristine. Like there's a lot of like weird, just kind of like how you survive as a human there. So yeah, hopefully that answered. It's kind of like a longer answer. Thanks, Mel.
Quick follow-up for Maris. I thought your artwork is such a pleasure, and I was just wondering, this book you're talking about, I have no idea how this stuff works. Do you have any idea of how long, like when it would be?
That's a great question. How long is this book going to take? Scientifically, I actually do, I look at each project that I've done and I kind of break down how long it takes for me to do a book. The first book took 5 years because I was working full-time, not on comics. But usually books take me about 2 years. This one's a little different because of the exploration aspect. There's a lot more scientific information in this book that have been in any of the other books. The other books kind of skew towards middle grade or late elementary school. The Antarctica book, I would like it to be understandable by that age group, but I kind of want to reach a little bit more towards high school and kind of just like general science engagement. So the research aspect of this book is going to push it a little bit longer. I would like to have it out by 2019, which means it has to be done by the end of 2018. It's about a year between when a book is completed and it comes out, which is like the longest waiting game. It's so hard. But yeah, this is a bit of a different nature. All of my other books have been, for the most part, just book research and interviewing people over the phone and not actually traveling to the place that I'm writing about. So let's hope it gets done by the end of 2018. Just because it'd be nice to have it out as soon as possible.
This is in relation to your comparative genomics and proteomics studies. Sorry. So have you found by any chance the presence of retroviral DNA, like junk DNA that may influence this pathway? And I don't know, there's something in my head, but I don't really know how to tell you. But since we basically evolved from viruses, according to several scientists out there, I was just wondering if you have found, even by mistake or by accident, those little bands, right? Oh, there's retroviral DNA in these samples in the different animals that you guys are choosing to study, like sheep versus seals, maybe versus human beings. And if that has anything, if you can find like a correlation between, I don't even know how to explain it, but it's in my head.
Are you talking about lines in science, like long and short interspersed non-coding elements in the genome and other intergenic stuff? Yeah, the question is, what could be in the genome that's not coding for proteins, but tells us a little bit about the evolutionary history?
There are viruses that have something to do with this pathway, and we can establish a correlation between the different species by having that same DNA in all samples. So I've seen studies like this where they have established that correlation, and how retroviral DNA has influenced significantly all these pathways that we're studying in different animals.
Yeah, I would answer that by saying that the genomic analysis is still in its early stages, so we're focusing mainly on protein coding regions and haven't gotten as far as retroviral elements in the genome. And one of the issues with these genomes where you sequence just one animal and then you have to align that against all other mammals that are known, genome sequencing has become much less expensive, but the quality is not the same as the billion-dollar human genome that we did a decade ago. And a lot of those retroviral elements are repeats, and they're masked in these assemblies, so they're sometimes hard to pick out. So I think that's the kind of thing that's not the low-hanging fruit, and that's the phase of the assessment that we're at the moment. But I think it would be great to get to that as we go forward. Yeah, the question is about whether we're able to look at the EKGs of these animals while they're underwater, and that is actually a topic of hot scientific debate at the moment. There's a number of warring papers that are talking about how whether or not parasympathetic and sympathetic innervation of the heart are at war because the animals are diving and exercising, and those two things are not, they shouldn't have the same impact on the heart. They should have the opposite impact on the heart. So that's something that's a discussion point, actually. Those traces are available. We've had digital EKG monitors on these animals for a few years now, so we're starting to get there.
Second part, is there any significant differences in the right ventricle in the diving seal relative to other mammals, terrestrial?
In terms of the anatomy of the cardiovascular system, that's the question. Yeah, the heart is much more compliant in these animals. It's because they have such a huge difference in cardiac output and stroke volume between diving and surfacing. But in terms of the right ventricular anatomy, I'm not sure.
Have you ever tried to measure pulmonary pressure?
So people have tried to measure pulmonary pressure, pulmonary arterial pressures, but they're zero because the lungs are collapsed. So it's a tough measurement to take. So we've been a bit stuck by technology.
Last question.
All right, last question.
So I know a lot of what you guys have been looking into has been kind of situations where they're more predatory. Have you guys ever come across any situations where they've been in more of like a prey situation where their heart rate might be a little bit higher, and how that might affect the nitric oxide levels or anything like that? I don't know if you guys would know that. I just was wondering if there's any difference between predatory prey situation.
Yeah, we started, we collect them on the surface where they're
really isolated. I didn't know if there's
and
anything you came across.
even those studies where they outfit the seals with computers, I don't think you even know what they're doing, right? I mean, you see a difference in heart rate, but is it because he's being chased by a predator or because he sees this tasty little fish? So
Yeah, the region that we work, even the animals that have been carrying critter cams in addition to the physiological monitors, there just aren't predators that we see. That's a really interesting question.
we'll stick around for a little bit longer. If anybody has any more questions, feel free to come down and ask us. Also, if you go to the website and the Instagram, you can always reach us through those. Thanks, guys.